Hello everyone. Today we see about the Endospore staining technique, its principle, procedure, result and difference. Now let's see about the introduction of Endospore staining. The Endospore staining method was published by Donner in 1922 and then Donner's method modified by Schaeffer and Fluten in the year 1933 to make the process faster. The endospore staining is a differential staining which the bacterial endospores stains selectively. Aim of the endospore staining. Endospore staining main purpose is to differentiate bacterial spores from the other vegetative cells. It differentiates the spore formers from non-spore former. Principle. Bacterial endospore are metabolically inactive and then highly resistant structure produced by some bacterial as a defense strategy against the unfavorable environment condition. The bacteria can remain in this suspended and they can germinate and become favorable and return to their vegetative state. Here, differential techniques are used to distinguish between the vegetative cells and the endospores. It has metabolically inactive structure and resistant to physical and chemical damage. And now see what are the reagents are used to do this technique. In this staining involved mainly four method of stains to give the culture color, fixing the bacterial culture in the slide and decolorizing. First, primary stain. Malachite green, it acts as a forced into the spore by steaming the bacterial emulsion. The malachite green was water soluble and has a low affinity for cellular material. Water is used to decolorize the vegetative cells and it removes unwanted stain. Mordant. Use of steamed heat. It is used to force the primary stain malachite green into the capsule covering. Safranin acts as counterstain. Any cells which have been decolorized at the end of staining, vegetative cells will be pink and endospores will be dark green. Spores shape may also be of diagnostic use. Spores may be spherical or elliptical. Material required: glass slide, inoculation loop, microscopic slide, bacterial culture like Bacillus species or Clostridium species, malachite green dye, water, safranin, microscope. Prepare a smear of culture on a clean slide. The organism on a glass slide is fixed by heat and air drying. Apply malachite green, the primary stain. Now allow the slide and then stay over the heating water bath for 5 minutes. Remove the slide from the water bath and gently wash it under running water. During 1 minute, coat the counter stain with 0.5% safranin. Examine for endospores on the slide by looking at it under a microscope. Vegetative cells range in color from brownish red to pink, whereas endospores are bright green. Result: endospores are appear green, vegetative cells red or pink. In this picture, we clearly see the endospores are in green, vegetative cells are in brownish red to pink. Some of examples: positive Clostridium prefungius, Clostridium botulinum, Clostridium tetani, Bacillus anthracis, Bacillus cereus, etc. In negative, E. coli, Salmonella species, etc. Now we see about the difference between vegetative cells and endospores. Surface coat. In vegetative cells, murine, cell wall polymer, general gram positive. In endospores, Dense pore coat cortex core wall of peptidoglycan. Microscopic appear in vegetative cells non-refractile, endospores refractile. Bacterial cells. It is the active form of bacterial cell, can be considered as the dormant form of the cells. Action of enzymes. In vegetative cells present, in endospores absent. Resistant to heat. In vegetative cells it is low and in endospores it is high. Resistance to acids, chemicals, radiation. In vegetative cells it is low, in endospores it is high. Synthesis of macromolecules. In vegetative cells present, in endospores it is absent. Calcium diploclolin acid. It is not seen and in endospores seen in core. Sensitive to lysosomes. In vegetative cells, yes. In endospores, no. Are they sensitive to staining and dyes? In vegetative cells, yes. In endospores, no. Thank you for watching.